This next set of slides looks at how genes can be regulated using various proteins and control of the other protein products. And this work was primarily done in bacteria, and that's what we will be looking at the moment. The genes that we will be looking at in E. coli can be described as catabolic genes, where they produce a protein product that degrades a substrate. And these genes are normally off, and we can turn them on. They are inducible. There are also genes that are anabolic, and these genes make a particular product, biosynthetic genes, and these genes are normally on, but if we already have that product in the cell, then we can turn this gene off by repressing it. These genes are therefore negatively regulated or positively regulated, turning off or turning on. And the whole reason these work is that there are a series of DNA binding proteins that help or hinder RNA polymerase here to do the transcription. So here is a cartoon showing negative regulation with a repressor protein. What we have here are two strands of DNA, so this is the double-stranded DNA right here. And this region, this yellow region here, is the promoter with the -10 tartar box in there. This molecule here is a repressor protein, and its job is to block the -10 area of that promoter region so that RNA polymerase, here's RNA polymerase, it cannot get in there to do any transcription, so there ends up being no transcription. So if we want to transcribe this gene, we have to remove this repressor protein, and the way we do this is by something called an inducer. This inducer molecule grabs the repressor, and they come together and they make this molecule over here. It's the inactive repressor and inducer together, and thereby that frees up this -10 tartar region of the promoter. And so RNA polymerase can now come in and transcribe that particular gene. So that's inducible transcription. A repressor protein binds the box, it is removed by an inducer, allowing RNA polymerase to bind the promoter to transcribe. There is also repressible transcription, which is the opposite. Here is the -10 tartar area of the promoter. RNA polymerase, of course, can come in and bind there, causing transcription, and so the gene is normally on. If we want to turn this off, we have a repressor protein. Here is a repressor protein here on the left. It's called an aporepressor because it's inactive at the moment. To make that aporepressor active, we have to add it to a co-repressor. And so the co-repressor and the aporepressor get together to make this active version, which then blocks the promoter region here, and so you get no transcription. So in repressible transcription, it's normally on, RNA polymerase can bind, but if you have a co-repressor, then together with the aporepressor, we can block the tartar box and to turn off the transcription. So the original work was done by two gentlemen called Jacob and Monod in France, and they studied the E. coli lactose operon. Now an operon is a series of genes that are linked together that perform a common function. So this diagram here, this is double-stranded DNA. Here are the two strands of DNA. There are 3 genes in this operon. There's Z, there's Y, and there's A, and they're all linked together, and they're all controlled by a single promoter. So here's the transcription start site here, and here's the promoter region, and what we have near the promoter region is something new. It is called an operator region, and this is going to be the binding site for the repressor protein. So this binds the repressor proteins. The gene that makes the repressor protein is called, in this case, the I gene, the Lac-I gene. It makes the repressor protein, and that is its transcription start site over here. It's a completely separate gene to the rest of the operon. So what does the Lac-operon actually make? Well, the Lac-operon makes a series of enzymes to degrade this sugar here. This is lactose. It's normal sugar that you would find in milk. And lactose is a disaccharide with a beta-galactosidase linkage, that's this bond here. And what's going to happen is it's going to break this disaccharide into two monosaccharides called galactose and glucose. And the enzyme is beta-galactosidase, and this is a product of that Z gene. So if we then turn on this particular gene, we see that here's the DNA across the top, here's the messenger RNA that is made, and here's the I gene. Remember, the Lac-I gene makes the repressor protein. The repressor protein will normally bind to the operator region and block any transcription. But in the presence of an inducer molecule, it will stop that binding and allow transcription to occur. So now we have a messenger RNA molecule with three genes, Z, Y, and A, all in the same messenger RNA, and producing three proteins down here. The first of which is this beta-galactosidase that cuts up the lactose into those two monosaccharides. So we will look at the two possible scenarios here. Here is the case where the repressor protein is made and it binds to the operator and you get no transcription. And so when the repressor protein is bound, RNA polymerase cannot come in and bind, and therefore you do get no transcription. If on the other hand the inducer molecule is present, then it will bind with the repressor protein to inactivate it, which then goes away, which then frees up the promoter region, allowing RNA polymerase to bind and therefore allowing transcription. And so you get expression of these three genes, which means that you can then get lactose broken down. So what would happen if not only lactose was present in the media in which E. coli is growing, but also, say, glucose as well? Which sugar, glucose or lactose, would the E. coli have a preference for? And this concept is called dioxic growth. And dioxic growth shows the preference of glucose over lactose given the choice. So we're going to look at the molecular mechanism by which this glucose preference is established. Now you probably know that if you have high levels of glucose, then that means high levels of ATP, because glucose goes through the glycolysis and Krebs cycles and electron transport chains to make high levels of adenosine triphosphate. What this also means, though, is that you get low levels of something called cyclic AMP, which is the breakdown product of ATP. So ATP and cyclic AMP are inverse to each other. If you have high of one, you have low of the other. And here's a diagram of cyclic AMP. So if there are high levels of glucose in the cell, you're going to have high levels of ATP, which means you're going to have low levels of cyclic AMP. The converse is also true. If you have low levels of glucose, you will have low levels of ATP, but very high levels of cyclic AMP. And so it is this cyclic AMP then that indicates the level of glucose in the cell. So we can use these levels to establish dioxic growth. So we're going to introduce another set of molecules involved in dioxic growth. And in this particular case, here is RNA polymerase, and it is bound to the promoter. But RNA polymerase can't bind on its own. It needs the help of another protein. And that protein is called CAP here. And CAP stands for catabolite activator protein. And this catabolite activator protein stabilizes RNA polymerase such that it can transcribe and produce Z, Y, and A messenger RNA. But CAP itself cannot bind on its own. It also requires cyclic AMP. That's the cyclic AMP from the previous slide. And so if you must have high levels of cyclic AMP, which will allow CAP to bind, which will then allow RNA polymerase to bind. So what this means is you must have high levels of cyclic AMP, which of course means low levels of glucose in the cell, to make all this bind and to get transcription. And so in this diagram we combine both the cyclic AMP and the cap molecules with the repressor molecule to show how transcription can be affected in the lac operon. And so in this case the repressor is present, which must mean that there is no inducer. And cap is also bound, which must mean there is high levels of cyclic AMP, which must mean there are low levels of glucose. So even though there are low levels of glucose, there's no inducer present to turn this on. In this bottom diagram we see the same system, except that the inducer is present and it has removed the repressor protein, therefore allowing RNA polymerase to transcribe Z, Y, and A genes. So what is this inducer? Well, as it shows here, this inducer is lactose. It is the compound upon which these proteins, the produced proteins, are going to act.